Hello, good afternoon. Thank you for joining today's live webinar, which will be presented by our guest speaker, Arun Radhakrishnan, titled Cellular Signaling by Cholesterol. Arun, if you'd like to share your slides while I do the introductions. So I'm Bill Griffiths from Swansea University, and I'm hosting today's seminar. The webinar will last around 45 minutes with 10 minutes for questions at the end. The seminar is being sponsored by Cayman Chemical, and we thank them for their continual support. And now a few words about our speaker. So Arun is at the University of Texas Southwestern Medical Center in Dallas, where he studies how cholesterol controls various cellular signaling pathways. Arun was trained in physical chemistry of cholesterol by Hardin McCall, and then cholesterol biochemistry by Mike Brown and Joe Goldstein. Arun, the floor is yours. We look forward to hearing the seminar.
Okay, thank you, Bill. Thank you so much for inviting me to participate in this webinar series. Okay, thank you again. So our interest for many years has been to understand how cholesterol acts as a signaling molecule to control many pathways. And in the next few minutes, I'll highlight a few of our findings. Okay, so as you all know, cholesterol is this waxy solid that is virtually insoluble in water. So the only way it can exist in cells is by being shielded in the lipid bilayer of cell membranes, where it imparts special properties to the bilayers to allow the functioning of membrane proteins and also to tune signaling pathways. Because of this importance, its levels are tightly controlled, and we have spent quite a bit of time, quite a bit of effort to understand how cells control their levels of cholesterol. So thanks to the work of many scientists over the past several decades, we know a lot about the machinery that regulates at least the production of cellular cholesterol. This regulation centers on a family of transcription factors called sterol regulatory element binding proteins, or SREBPs, that were discovered by Brown and Goldstein 30 years ago. And one of the isoforms called SREBP2 controls all the genes for cholesterol synthesis and also for the LDL receptor that orchestrates cholesterol uptake. In a great example of negative feedback, when cholesterol, the end product of these processes, builds up, it inhibits the activity of this SREBP2 transcription factor, and cholesterol synthesis and uptake are reduced. So how does cholesterol do this? The key is a membrane protein called SCAP. So in the endoplasmic reticulum membrane, the ER membrane, SCAP forms complexes with the SREBPs, which I just mentioned. And when the cholesterol levels are low, when you're in a cholesterol deficient state, the SCAP transports the SREBPs from the ER to the Golgi, where the SREBP is cleaved by two proteases, and that releases the SREBP's N-terminal transcription factor domain, which can then go to the nucleus, where it activates the genes for cholesterol synthesis and also the LDL receptor. So the overall effect is an increase in the production of cholesterol. So as a result, the cholesterol levels in the cell rise. It rises in all the membranes of the cell, and eventually even in the ER membrane. And so when this level increases above a threshold, the excess cholesterol binds to SCAP, and it changes its conformation so that it can no longer transport from the ER to the Golgi. As a result, the SREBP is trapped in the ER. It is not cleaved by the proteases, and its active transcription factor domain is not released to go to the nucleus to turn on genes. So as a result, the cholesterol production decreases. Right, so this is the feedback loop that maintains cholesterol within tight limits. So how sensitive is this process? So by measuring SREBP's activation status in cells, that's on the y-axis here, as well as the cholesterol content of purified ER membranes from those same cells, we can get an idea of the sensitivity. The results are striking. below a threshold concentration of about 5 mol% cholesterol in the ER membrane, SCAP is transported from the ER to the Golgi and the SRBPs are activated. So you have this high activity here. Above this threshold, SCAP's transport is abruptly terminated, and so the activation of SRBPs is shut down. So you get this switch-like response. And we've spent the last decade trying to understand the molecular basis for this switch-like response. So let's take a closer look at SCAP to see how it may be doing this. So SCAP is this large membrane protein. It contains 8 transmembrane helices connected by 7 loops. Some of them are large, some of them are small, and about half of the protein extends into the cytosol as a C-terminal domain. So working with Brown and Goldstein, and now since then, without them, we have painstakingly mutated pretty much the whole protein, right? And that molecular dissection, if you will, has allowed us to define functional domains of SCAP. So for instance, we know that the C-terminal domain binds the SREBPs, the CTD. This loop 6 here, it binds the COP2 proteins that package the SCAP and the SREBPs into transport vesicles to move it from the ER to the Golgi. This blue transmembrane bundle binds the ER retention factor, called the INSIG proteins. And these two loops in the lumen of the ER, these 2 luminal loops, they bind each other. Interestingly, the cholesterol binding region in the protein is not in the transmembrane region, but rather in a loop, in one of these luminal loops that projects into the loop. Okay. And then in the last couple of years, we and others have even obtained cryo-EM structures of SCAP that have at least verified SCAP's architecture that we inferred from the biochemical studies. But all of these advances have told us nothing about how SCAP senses these small changes in ER cholesterol. Like, how does this switch-like response arise? And one of the reasons for that is that we have focused almost exclusively on the protein SCAP, but that's only one half of the interaction that drives this sensing mechanism. The other half is the ligand, cholesterol, which is in the ER membrane, where it's buried amongst all the phospholipids that it's around. So how does SCAP's extramembranous loop here detect the cholesterol in the membrane? So to answer this, we need to expand our focus to include not only SCAP, but also how cholesterol is organized. So obviously this is difficult since membranes are liquid. They do not have long-range structure like a protein does. So we've approached this problem in many ways. The most fruitful of these approaches has been the use of bacterial cholesterol-binding toxin proteins. Okay, so I know it's kind of weird, but I'll tell you how we got there. So most bacteria don't have cholesterol, right? But many of them, they secrete soluble toxin proteins that bind cholesterol in the membranes of target cells. So one family of such proteins are called the cholesterol-dependent cytolysins. There's more than 20 members of this family. The crystal structures of 6 of them are shown here. They all share the same architecture, and they're with a cholesterol binding domain shown in gold here. They all contain a conserved hexapeptide, which is important for binding to cholesterol. All right. So the 2 best studied members of this family are perfringolysin O and anthrolysin O. And both perfringolysin O PFO and anthrolysin O ALO, and all the other members of this family act in the following way. They bind to membranes only when the membranes contain cholesterol through their domain 4s, this D4. And then they oligomerize, and then they form large pores to lyse the cell. Since bacteria don't have cholesterol, the toxin doesn't harm itself. It only helps it target the cell that it wants to target. So we're not microbiologists, but we were interested in the system since the protein is binding cholesterol at the membrane surface. reminiscent of SCAP's extramembranous loop 1 binding membrane cholesterol. So we looked at this in more detail. I had shown you this top curve before. This is the response of SCAP to the amount of cholesterol in the ER. So we measured the binding of these toxins to those same ER membranes. And we noted a similar sigmoidal dependence of the binding of these toxins to ER membranes. And the break point in this sigmoidal curve occurred at roughly the same point where SCAP's activity was terminated. So there's no sequence or structural similarity between SCAP, this eukaryotic membrane protein, and PFO or ALO, which are these soluble secreted bacterial proteins. So this common threshold likely reflects a property of the membrane. So the way we think about it is that below the threshold of 5% in the ER in this case, most of the cholesterol is sequestered or held in an inactive form through interactions with other phospholipids and other membrane factors. And then above this threshold, some of the cholesterol becomes accessible. The sequestration capacity of the ER membrane is overwhelmed. And so this accessible cholesterol accumulates at the membrane where it can bind to sensors such as SCAP or to the toxins such as PFO or AL. All right. So we dug into these commonalities a bit further. So we screened a panel of sterols. The details of the sterols aren't important in this for right now. And we screened this panel for their binding to SCAP. Okay, this is a radioligand binding assay, competition binding assay, to test their ability to displace pyridium-labeled cholesterol from the binding site in SCAP. So we found some, the ones in red, that displace cholesterol from the protein, and then others, the ones in gray here, that don't. All right. So again, the details aren't important, but what is important is that when we tested the same panel for binding to this bacterial protein, ALO in this case, we got the exact same specificity. That is, the sterols that bound SCAP, the ones in red here, also bound to ALO, and the sterols that did not bind to SCAP also did not bind to ALO. Right? Okay. Perhaps most striking is that both proteins bind cholesterol, but not epicholesterol, which is a diastereomer that differs only in the orientation of this hydroxyl group here, right? So ALO is binding to cholesterol, which shows the same exact affinity and specificity as SCAP's binding to cholesterol. This is most unexpected. So we took a leap and we set up a high throughput screen to find a small molecule that would bind ALO as a surrogate for the much harder to work with membrane protein SCAP. So we screened about 300,000 small molecules for binding to ALO. And we took the hits and we subjected them to a barrage of counter screens and found one molecule that binds SCAP in a specific way, and it blocks activations of SREBPs. So this molecule, which we call UT59, it looks nothing like cholesterol, but it's much more soluble, right? Here's an example of how UT59 works in this Western blot here. So when cells are depleted of sterols, SCAP triggers the cleavage of SREBP from its large precursor form to its process nuclear form, and this this cleavage, which happens because SCAP is transporting SREBP, gets blocked by adding increasing amounts of UT59 in the same way that adding a sterol like cholesterol would. Right. So this happens for both SREBP2, which is what I showed you before, controls the genes for cholesterol, as well as SREBP1, which controls the genes for triglycerides and fatty acids. Right. Consistent with this effect on SREBPs, lipid synthesis in cells goes down when you treat them with UT59 as expected, and also the UT59 also works in lowering SREBP activation in mouse livers. So we're not aware of any compound that has this specificity and potency. So we're hopeful that this reagent can, both serve as a tool to understand SCAP SREBP better, and perhaps even provide a starting point for a scaffold to go for a drug-like compound that could be useful to study animal models. So who would have thought that a bacterial protein could, give insights not only into the mechanism of cholesterol sensing, but also help us find an inhibitor. So we wonder, what else could these bacterial toxins tell us about cellular cholesterol? So the SCAP-SRBP pathway that I described to you, it's in the ER membrane. The ER membrane only has a small fraction of the cell's cholesterol. Some estimates put it at, less than 10%, about 1% or so. Most of the cell's cholesterol, the large majority, is in the plasma membrane of the cell. And in the plasma membrane, cholesterol makes up almost half of the lipids in that membrane. So we decided to use these bacterial toxins, ALO in particular, to study this cholesterol-rich membrane. So we wanted to study live cells. So we first engineered a version of ALO, a truncated version of ALO that would bind and sense the cholesterol, but not without the side effects of lysing the cells. So we call this truncated sensor ALO-D4. Okay. So the binding of ALO-D4 to mammalian plasma membranes from a variety of culture cell lines is low until you reach a threshold of about 30 mol%, above which the binding increases sharply. So this threshold is reminiscent of what we saw with the ER membranes, except it's at a much higher concentration. The ER membranes, it was at about 5%. So the higher sequestration. So what is behind this higher sequestration threshold? It's largely because of sphingomyelin, which is a phospholipid that's present at high concentrations in the plasma membrane, but is largely absent from the ER. Indeed, treating the cells with the plasma membranes with sphingomyelinase, an enzyme that degrades sphingomyelin, liberates previously sequestered cholesterol and makes it accessible, shifts the curve to the left. So the way in which sphingomyelin sequesters cholesterol can be explained through a simple model where sphingomyelin forms complexes with cholesterol. There's much evidence for these complexes from model membrane studies from many years ago, but such complexes have not been directly observed in cell membranes. Thankfully, recently we were lucky in when we were screening other toxins. This ALO-D4 senses this accessible cholesterol, if you will. We were screening other toxins, and we found a fungal toxin called osteolysine A, or OLE-A. This toxin is interesting. It binds only to sphingomyelin cholesterol complexes. So it's a sensor for the sphingomyelin sequestered form of cholesterol. The OLE-A doesn't bind free sphingomyelin or accessible cholesterol, and this ALO-D4 doesn't bind this form of cholesterol. So we have distinct sensors for these two forms of cholesterol, if you will, in the membrane. Right. So using these two sensors, ALO-D4 for the accessible pool, OLE-A for the sphingomyelin sequestered pool, we can now quantify these different states of cholesterol in the plasma membranes of cells. So in culture cells grown in lipid-rich media, like 5% FCS, if you will, so there's lots of cholesterol, we find that about a third of the plasma membrane cholesterol is accessible. That's this green pool here. Another third is sequestered by sphingomyelin, and then the remaining third is sequestered in some other way, right? So when you deplete cells of cholesterol by growing it in a lipoprotein deficient serum, then what's interesting is that the cholesterol goes down as expected, but only this accessible form goes down. This red and purple forms stay roughly the same. We observe the same. So we go from culture cells to primary cells from tissues such as the mouse spleen or human red blood cells or mouse hepatocytes. We found a different result than what we found in culture cells. In these cases, the accessible cholesterol pool is barely detectable. So this may make sense since we're beginning to appreciate that this accessible pool of cholesterol is a potent signaling molecule in many pathways. So confining the signaling potential of cholesterol to just this rare pool, it could be a way for the cells to maintain large amounts of cholesterol, but still allow for small changes to trigger signaling. So we're beginning to better understand some of the signaling roles of cholesterol in the last few years. So here's a few of them. So this accessible cholesterol from the plasma membrane, we can show that it transports to the ER where it binds to SCAP, as I talked about, to lower lipid synthesis. It can also accumulate in the cilia, which are membrane projections from the plasma membrane, and bind to a GPCR protein called smoothin, which is part of the hedgehog signaling pathway to activate cell growth. Not all is so these are these roles allow the cells to maintain lipid homeostasis and also to grow. Not all is good. This accessible plasma membrane cholesterol can also be a vulnerability, as many bacteria, including PFO and ALO, obviously they're binding this pool. These bacteria and even some viruses, they exploit this pool to promote infection. So working with our colleague in microbiology at our school, we recently, Neil Alto, we recently uncovered an innate immune response by the host cells, which rapidly and selectively removes the accessible cholesterol from the plasma membrane to provide protection. So let me tell you just a little bit about that. So when infection happens, you get the interferon response, which turns on many genes, these so-called interferon-stimulated genes. And one of them is an enzyme called CH25H, discovered by, blown first by David Russell, who LIPID MAPS knows all about. And this CH25H converts some of the cell's cholesterol to an oxysterol called 25-hydroxycholesterol. So this 25-hydroxycholesterol can enter an infected cell, and it blocks both the invasion of the bacteria as well as the spread of the bacteria from one cell to the other. And it does this by modulating the accessible pool of cholesterol in cells. Okay, so upon infection, as a model, if you do LPS addition, the induction of the CH25H gene is quite rapid. You get this sharp spike. It's a paper by David Russell from a decade ago now, where the CH25H gene mRNA goes up after 4 hours and then it declines. And if you use our probes, this ALOD4, to measure the level of the accessible form of cholesterol in plasma membranes, you start off with a robust amount, and upon 25-hydroxycholesterol treatment, by 1 hour, this is largely gone. By 4 hours, it's mostly gone. So, and that confers protection against the bacteria, which need this accessible cholesterol to enter cells and to spread. We're still working out those details. But how does 25-hydroxycholesterol lower the accessible pool of cholesterol? So, as it's been well known that 25-hydroxycholesterol affects multiple cholesterol homeostatic pathways. So 25-hydroxycholesterol is made from cholesterol, and then it feeds back to lower cellular cholesterol by degrading HMG-CoA reductase, the rate-limiting enzyme in cholesterol synthesis, and also blocking the activation of the SRBPs that I just mentioned about. So these two processes lower the cell's cholesterol, would lower the cell's cholesterol, but they would take time. The SRBP is a transcriptional process, so that would take hours to manifest. 25-hydroxycholesterol also activates the LXR pathways, which exports cholesterol out of cells. This may also take time because this could also be a transcriptional response. And it also stimulates the activity of an enzyme in the ER called ACAT, which esterifies cholesterol and puts it into lipid droplets for a storage form. Okay? So we have now worked out these pathways. When we find that 25-hydroxycholesterol, remember, it it acts quickly. By like an hour, it can remove the accessible cholesterol from the plasma membrane. So we think that fast response, the rapid depletion, is largely due to its activation of ACAT. And then the sustained, long-lasting depletion of cholesterol comes from its effects on the other pathways that lower cholesterol. Okay, so that's a little insight into what we have learned using these toxin sensors about one signaling pathway. So I want to tell you in my in the last 10 minutes or so about where we're going next, or one of the directions we're going in next, and that is, can we use these toxin sensors not just to probe the levels of these different forms of cholesterol, but can we actually see these lipids in these different forms in a membrane? Okay, so we have made progress with, I mean, these are soluble proteins, ALO, D4, and OLEA, so we can crystallize them. We have very high resolution structures, but we don't know how they interact with lipids in a membrane. This is just grafting the crystal structure onto a membrane interface. We don't know what this interaction looks like. So we've been spending a lot of time trying to get at that answer with both ALO and OLEA. We have made some progress with OLEA, so I'll tell you about that next. So OLEA is a small protein. It's 15 kilodaltons. So it's not, so it's great. We were able to get crystal structures out of it, but then when you want to get structures on a liposome by using cryo-EM analysis, it didn't work because the protein was too small. But thankfully, OLEA acts with an additional cofactor. It's called another protein called PLI-B. The details aren't important right now, but with this cofactor, OLEA can assemble into large membrane-bound oligomers, which can be megadalton in size, right? So that may be a better candidate for cryo-EM. So let me show you that. So OLEA, it's a soluble protein. It binds to sphingomyelin cholesterol-containing membrane, dimerizes, and that dimer provides a binding site for this second protein called PLI-B. That comes and binds here. Then it undergoes, the PLI-B undergoes conformation changes. Magic happens, it forms an oligomer, and then it makes a pore. And the pore has 2 OLEAs and 1 PLI-B. That's the basic unit, and there's 13 of those. Okay, so it forms this 13 mer, if you will, and that whole complex is 1 megadalton. Okay, all right. So we went for this complex to get at the structure. So both of these proteins, their crystal structures have been solved in solution. You can purify them quite easily. You get large amounts of it. And then we took those 2 proteins, and then we made, because the OLEA is binding sphingomyelin cholesterol in liposomes, we just started with, putting in as much as we could. So we made a 1-to-1. We made liposomes containing just 2 lipids, 18:1 sphingomyelin and cholesterol, 1-to-1 molar ratio, right? So we make these liposomes, and then we add the proteins, incubate them, and we put it on a cryo-EM grid. This is what we see. Here are the liposomes, and you can see these pores, if you will, on there. Right? If you zoom in, you can see this 13-fold symmetry. In a side view, you can see the protein going through the membrane. Okay? So, okay, this is good. We collected a lot of these images, and then we processed them to get a molecular model. So that's what it looks like. That's what that looks like. So here's the membrane. There's the OLEA in blue. And there is the PLI-B protein in green. And this is the side view. And then if you turn it around, it's going through the membrane to make this hole here. So from the top, you can only see the PLI-B. The OLEA is underneath here. You can maybe see a little bit of it sticking out here. Okay, resolution is not bad. Overall resolution is about 2.6 angstroms, which is not bad. But remember, we care about this interaction, this blue with the membrane, right? The OLEA with the membrane. So let's look at, whenever you see people reporting, that the resolution is 2.6 angstroms, I mean, great, but usually the resolution is best at parts of the protein you don't care about, and that was the case for us as well. So here is that same structure colored by resolution. Okay, so the overall is 2.6, as I said, and so the blue is what we're going after. Around 2 angstroms is where, for the pore, that's what we see. But where the all-EA is touching the membrane, the resolution is much worse. It's around 5 or 6 angstroms. So it's 5 or 6 angstroms. You can barely see the protein, let alone any lipid that it may be binding to, right? So that wasn't going to get us where we wanted to go. So we thought, well, we got to try something. So we went back to this picture and we thought, well, maybe the job of all EA is to bring, is to bring this pore to the membrane, if you will, to start the process. It binds the membrane and it brings pli B to the membrane. And then maybe once it makes a pore, its job is done. And so maybe it's no longer as tightly bound to the membrane and it's more flexible. And maybe that's why we're getting a lower resolution. So if that's the case, we thought, well, if there's some way we can get the oligomer to form, but then arrest the process before it makes the pore, then maybe we would still see then the oligomer would need to be bound to the membrane through all EA. So maybe that would help us, right? So we wanted to block this somewhere over here. Okay, so how do we do that? So we wanted to basically prevent the conformation changes in this green pli B protein. Thankfully, that's where the resolution was the best. Going back here, the resolution in the pli B section, that's this dark blue here, so it's 2 to 3 angstroms. So we had a lot of information for that state. So the pli B protein, it's a globular protein in solution. And then when it goes into the membrane, it extends. It undergoes this remarkable large conformation change. Let's look at a couple of pairs of amino acids. So like, for instance, if you look at this tyrosine and phenylalanine, which are within about 2 angstroms in the solution structure. But then when it's in its extended form in the membrane, it becomes more, they separate by more than 100 angstroms. Here's another pair of this histidine and glycine, which are also within about 2 angstroms here, and they separate by about 100 angstroms in the membrane-bound form. Okay, so we thought, well, maybe if we replace those, so the protein conveniently only has one cysteine, one native cysteine. We've mutated that cysteine to a serine, so that protein is still active. So we have a cysteine-less pli B. So we thought, well, maybe if we take these close proximity amino acids and mutate those to cysteines, maybe we could, if they formed a disulfide bond, then maybe that would arrest this stretching out of the protein. So we tried many different pairs of amino acids, and in the end, these 2 pairs, mutation of these 2 pairs worked. So let me show you the data for that. So here is using pli B with 4 mutations, these amino acids to cysteines. So the wild type pli B makes a pore. That's just the hemolysis assay for pore formation. But then we call it this transmembrane hairpin lock protein. TMH lock protein does not. If that was because of disulfide bond formation, then you would think if you added a reducing agent, you would break the disulfide bond, and then you would be able to make pores again, and that's what happens. So this TMH lock protein can make pores if you add DTT to it. Okay, cool. So that was one way to arrest the pli B in a non-pore forming, right? Okay, so that was a start. Another way another strategy we came up with was there are 2 glycines which are adjacent to each other, and they are important for this unfurling process. They're conserved in all of these pore-forming proteins. And some earlier work had suggested that mutation of these glycines prevented pore formation. So we thought, well, maybe these also would arrest arrest the pore formation in a way that's interesting to us. So we mutated those two glycines to valines, and again, the wild type makes pores. This glycine to valine mutation doesn't. Good. So then we went ahead and did cryo-EM analysis with the same OLE wild type, but with these mutant forms of PLI-B. Okay, so the wild type gives you these pores that I just told you about, and what we were very excited by was that these two mutant forms of PLI-B also formed oligomers, right? Okay, I mean, in this one, if you look at this and that, it doesn't look that different, but I'll show you that it's not making a pore, and that's what's getting us to where we want to go. All right, so okay, so cool, they're making oligomers. Let's see what happens. The PLI-B wild type makes pores. The mutants don't, as I showed before. And importantly, it doesn't affect, the mutations don't affect the specificity of the system. So the OLE binding to just sphingomyelin cholesterol is still maintained in these mutants. Okay, so this here is just showing again that you get binding to liposomes containing sphingomyelin and cholesterol 1-to-1, but no binding to sphingomyelin alone liposomes or to sphingomyelin plus epicholesterol liposomes. That's that diastereomer where that hydroxyl is going the other way, right? And then if you make liposomes with a glycerophospholipid, here's a di-18-1 PC with cholesterol or epicholesterol, you get no binding. Okay, all right. So okay, so we went ahead, did the same thing. We solved the structures, and here's what we got. Here's a wild type again. Here's a top view. It's making a hole. That's this white hole in between, in the middle there. And then these two forms of PLI-B, they it's black. They're not going all the way through. If you take a side view, you see the wild type goes through the membrane, and these two forms of PLI-B, they do not penetrate the membrane. Okay. All right. So I talked a lot about PLI-B, but remember, our whole goal was to get this blue part to higher resolution, right? So did we do that? So here's showing that same resolution, the same structural map colored by resolution that I showed you before for the wild type. Recall that resolution was high, this beta barrel part going through the membrane, but not so good for the OLE. But then when we made that disulfide lock, it made this 5-6 angstroms get closer to 4. Okay, better, but still not that great. But then this glycine hinge mutation really made this better. So where we are at the membrane is now 2 to 3 angstroms. So that's cool. So we focused on that area. Like now let's look to see whether we can see the lipids. So here's a low pass, low filter map of that. So you can see the OLE. You can see this connection to the membrane. And if you look closely there and you refine this data, you can see the density, which is modeled by a sphingomyelin and part of a cholesterol. I'll show this in a little bit more detail in the next one. So here is the map again. You can see the phosphocholine and then the ceramide backbone, and you can see it's supposed to be a movie. I'm trying to see, but okay, it's not playing, but that's okay. But then here's the cholesterol steroid nucleus, and you can see a connection between the hydroxyl group of the cholesterol and this amide nitrogen on the sphingomyelin. So there's this hydrogen bond that's been proposed from calorimetry studies in the past and other biophysical measurements. And here's a visualization of that in a membrane. And you can see the beginnings of both acyl chains of the sphingomyelin, and then and then the head group is kind of bent over, bound to the cholesterol. And this could be how it forms a complex and keeps that cholesterol in a sequestered form to prevent its function in signaling pathways. So that's kind of where we are right now in trying to get a biophysical view of the cholesterol states in membranes. It's worth noting that I've today talked about three different proteins that bind this accessible form of cholesterol. This is this ALO protein, smoothen the GPCR, and SCAP, the cholesterol sensor, and this all-EA protein that binds sphingomyelin cholesterol. Right. In all cases, the binding region in these proteins, shown in green in the different proteins, they're all outside the membrane, right? And but they all have to then kind of dip into the membrane, if you will, to some extent to bind their lipids of choice. So yeah, we keep going at this, trying to understand this better. So I thank you. I was told to keep this for 40 minutes, so that's where I am. So I'll stop here. I just thank my collaborators, Neil and Raj, who helped with the microbiology and the hedgehog signaling part, and then my trainees, not Mike and Joe there. Thank them for their support on the SCAP project, and my Simon and Jared, whose work I presented today. And I thank you for your attention.
So thanks, Aaron. That's a really, really great talk. And isn't it incredible to think that such an abundant molecule as cholesterol can actually be a signaling molecule? It's incredible, isn't it?
That is, yeah. It would make if you try to explain that, at one level it makes no sense, because you would be like, well, how would you ever turn the signal off if it's 50% of the membrane, if you will? But it's turns out it has these different strategies to keep it inactive, if you will, keep it inert.
Yeah, it's really interesting. So now we got some questions from the audience. So the first questioner is Scott Hansen.
Hi, great talk, fantastic. So a couple of things. I think probably the most important is, or not the most important, but maybe if I can just pick a couple. One would be like, how fast can this process actually happen? I asked that, like, it could be, even an hour is pretty slow, but have you really pushed it to see, like, if you add, say, 25 hydroxy cholesterol, will it signal, like, how fast could you actually block viral entry? Like, if you added it for 5 minutes, what would happen?
Okay, we have not done experiments like that to get at how fast it is, but it can, because a lot of the infection assays are, you do some treatment and then you measure something 20 hours later or 16 hours later, so it's hard to do really short-term infection studies. But you can, but we can look at, like, how fast does the accessible cholesterol level in the membrane change, and that happens quite rapidly. Within 10-15 minutes it can happen, probably even faster than that. It happens as fast as we can measure.
That is fantastic. So a really quick question, does UT59 work in the brain?
We don't know whether it crosses the blood-brain barrier yet. Its bioavailability is limited right now. It works great in cultured cells, and it works. The animal study I showed was an IP injection for 6 hours, so it's okay there. But it's
would it work on astrocytes? Do you think the main cholesterol producing in the brain?
I would anticipate it would work in astrocyte culture cell systems, but we don't know. I mean, it should work in the brain if we could get it there.
And then my last question is, so cholesterol changes the hydrophobic thickness of lipids. And so, for example, scrap protein that's in the ER, or even the ones that are on the extracellular membrane, if the membrane gets thicker, the helices would either have to stand up straight or somehow deal with the hydrophobic mismatch. Have you thought about that? And have you looked at the actual transmembrane parts to see whether they're like 40 angstrom, closer to, say, thick membranes with cholesterol, or are they closer to, say, a disordered type PC membrane, which would be closer to 30 angstrom?
You mean in the structures that I showed?
Yeah, like the structures. If you just look at how thick the transmembrane domain is, what do they look like they want to be in? Because if you have a thin transmembrane domain, say for example 29, 30 angstrom, that's going to have induce puckering or something to accommodate the hydrophobic mismatch. Or is it thick, like you would expect for a sphingomyelin cholesterol complex?
Right. So these kind of approaches we have used to using the protein to probe the membrane so far we've only been able to do it with a sphingomyelin cholesterol membrane, right? Because the protein doesn't bind to a PC alone or a sphingomyelin alone membrane. So we're limited in that sense. But now that we have this, we can do we can plan experiments to kind of get at what you're saying. So maybe we could make membranes containing increasing amounts of cholesterol to see whether we can change whether we can see differences.
But if you just look at the structure, how thick's the, like, does the
in this case, it's about 35 angstroms.
35. Okay.
Yeah, so
Interesting. And you think if it went thin, would it change conformation
it's
and
about,
then no
it's about
longer?
35 here, but the resolution isn't great in the membrane part, right? As you would imagine, but it's about 35.
Okay, let's have the next question. Thank you, Scott. Next question, Jason Miller.
Thanks. I am a cell biologist, so my lipid knowledge is limited. But I'm curious whether or not a cell type that I'm interested in, photoreceptors, have these membrane stacks that originate from plasma membrane, but the stacks then become fairly cholesterol poor, closer to 10% cholesterol in their membranes, and that stimulated in me, especially with how tightly the rheostat in the ER is set for cholesterol detection at right around 5% mole. Is there any differences among cell types in that rheostat in the ER, depending upon what types of percent composition of cholesterol the rest of the cell needs for various structures or plasma membranes? Or is across all cell types, is this 5% rheostat sort of set, and SCAP has the same sensitivity, despite maybe differences between cell types in their need for cholesterol in plasma membranes or other intracellular structures?
I see. So I'm sure that it's not going to be exactly 5% across all cell types, right? There's going to be differences for sure. We have established a lot of these principles in cultured cells, CHO cells, SV589 cells, and in there it's between around 5% or so. We've also looked at mouse tissue, hepatocytes from mouse livers. There also there is a threshold, maybe a little higher, around 8 to 10%, if you will. But so it's between around 5 to 10% in at least 2 or 3 different systems that we have looked at. Perhaps instead of trying to get at the exact number, maybe I think about it more like the plasma membrane has about 40 mole% cholesterol, and the ER has about 10-fold lower. So I think about it maybe more like that. So the ER membrane maybe is around 10-fold lower cholesterol concentration than the plasma membrane. And sure, there could be changes in the exact numbers, but but it's this difference in the concentration between the plasma membrane and the ER that's important.
Yeah, I think I understand that principle. I guess what I'm saying is that certain cell types may be unique in their cholesterol needs, and I'm curious whether or not some of that is regulated by the SCAP sensitivity in the ER, the rheostat, or regulated by the myriad other mechanisms that govern trafficking between different membrane structures.
That's true. You can also think about if you didn't have the ACAT enzyme, for instance, to take the excess cholesterol in the ER and put it into droplets, maybe those ER membranes would have higher amounts of cholesterol because they don't have a safety valve to get rid of the cholesterol, right? So and then as you said, if you have a very aggressive transport system or a lethargic transport system to move the cholesterol between the membranes, then that could also affect the amount of cholesterol in the ER.
Thank you very much.
Okay, the next questioner is Alison Ondros.
Great. Well, thank you so much, Arun. That was excellent. I had a quick sort of structural question regarding SCAP. So you have sort of the picture of SCAP in the membrane at the end where there's this obvious gap between the loop region and the membrane that's much farther than, say, the hydroxyl group at C3. So I was just wondering what you envision sort of for the path by which cholesterol in the membrane accesses that loop region, and whether you think there might be some sort of dynamic events that bring the loop region close enough to sort of sequester it.
Yeah, no, that's a great question. We don't know the answer to that. Our resolution of SCAP, again, the nominal overall resolution is about 4 angstroms, but in the area where the loop one is close to the membrane, it's much worse. It's around 5 or 6, like it is for the wild type LEA, and we can't resolve the whole protein. So we don't know those details yet. It is worth noting that if you prepare an expression plasmid expressing just the loop one of SCAP, then that recombinant protein is not soluble. It requires detergents for solubility. But if you make a fused protein where the loop 1 and the loop 7 are together, then that fusion protein is soluble. You don't need
that's interesting.
That's so if you wildly extrapolate from that, then you could think that maybe the loop 1 when it's not bound to loop 7 is bound to the membrane peripherally, and then it can scan between at least 2 states where it's bound to loop not or so. But yeah, we don't know. We're working on those things.
Oh, that's very interesting. I had the other quick question I can ask, ideally quick, is that what do you think the sort of rationale for this intermembrane this sort of location of the loop region is? Do you think that feeds into sort of the homeostasis signaling?
Oh, I no, you'll have to ask. You'll have to ask her why she did it that way. I would have maybe thought you would put the sensing system in the membrane, which has the most cholesterol, like the plasma, but it's not there. It's in this ER membrane, and then if it's in the ER membrane, you would have thought, well, maybe it should be in the outer leaflet where it would be receiving the cholesterol from the plasma membrane, but that doesn't seem to be the case. So, so I think I think the answer is I just need to come up with the hypothesis, and then whatever I think, just go with the opposite, because that's what
sounds like a plan. Thank you so much.
Okay, so next up is Dinesh.
I'm just wondering what is the driving force for the elongation of OLA protein into the membrane? Is it just because the presence of cholesterol and sphingomyelin, or what is the driving force to elongate OLA?
Okay, so the LEA doesn't change. The LEA is it looks like that in solution, and then in the pore structure it still looks like that. So the RMSD between the membrane-bound form and the solution form is very small. What is changing a lot is this PLI-B. That goes from this globular protein, like I showed, to something extended. And that is happening because of it's triggered by the binding to LEA. So the PLI-B, this is a lipid-independent event, if you will. It's an LEA-dependent event. Once it binds to LEA, its conformation changes, but it only can bind to the LEA when the LEA is bound to the membrane. There's mechanisms by which the PLI-B cannot bind to the LEA in solution. They only bind to it when it's on the membrane, and that may have to do with how the conformation of the LEA in solution is. Yeah.
Okay, thank you.
Okay, next up we've got James Thorne. James.
Yeah, hi. First of all, thank you for that talk. That was absolutely fascinating. So I wanted to ask about the therapeutic angle here. So obviously you've identified that LEA and PLI-B are essential for allowing the bacteria to sort of damage and infiltrate mammalian epithelial cells. Are these novel antibiotic targets, or are these already known and targeted through existing therapeutics?
Oh, I don't know of an existing therapeutic which is targeting these sensors. No. And as you know, these microbes have 100 different ways to kill a cell. This is one of them. So I don't know if that's the reason why, but I don't know if therapy that's targeting this angle, but that could be interesting to look at.
Okay, thanks. And then I was wondering as well, if have you got consistency between the structures that you've developed and sort of molecular docking or any sort of like atomistic simulations? Do you replicate the same interactions?
Yeah, we can. We have tried going at this step, this all-EA binding to the membrane by some molecular dynamic simulations. We haven't been very successful yet, and that's because our lipid box, as you know, can't be too big. It's too computationally intensive. But we would love to work with somebody to do that. No, we don't have that answer yet.
Okay, great. Thank you.
Okay, we're running out of time, but we've got time for perhaps one or most two more. So Gabriela Limos.
Hi, congrats for your talk. Thank you very much. I'd like to ask you about the role of the length of the sphingomyelin and the bounds specificity with OLIA, and if it does it have a role in the cholesterol metabolism. Thank you.
Okay. The specificity of the acyl chain length for binding OLIA is it binds best to sphingomyelins that are 18-1, 24-1, 18-1 to 24-1. The binding is 50% less when you go to a 16-0 sphingomyelin, and to lower ones, a 14 or a 10 is almost nothing. So it requires chain lengths of 16 or higher, if you will. And then perhaps that helps the sphingomyelin form van der Waals forces to interact with cholesterol. Maybe that's the reason why. I don't know.
Thank you very much.
Okay, so we have one last question here now. Last one from Ahmed El Montasser. Ahmed?
Yeah, I want just to ask if it's known the exact mechanism behind cholesterol lowering, what you saw by the bacterial toxin regarding the innate immune system. Is it only about the pathogen entry and that's related to cholesterol, or is by trapping the pathogen inside and reducing the pathogen egress?
Yeah, so good question. We don't have all the answers, but if you think about a pathogen like, say, perfringolysin or anthralysin, where it needs to bind to the cholesterol on the membrane to oligomerize and form a pore. In that case, it's perhaps straightforward to go, well, then if you just remove the cholesterol, it can't bind, therefore it won't harm the cell. But for other things like like viral entry or for intracellular pathogens like, say, Shigella or Listeria, where the entry seems to be determined by interactions with internalins or other protein receptors. There, it seems that the role of the cholesterol is not in the invasion step, but rather in the cell-to-cell spread step. How, we don't know, but for cell-to-cell spread to occur, membrane protrusions need to form, and it seems like and what we find is that in the absence of accessible cholesterol, the number of protrusions that form are dramatically reduced, and maybe that's why that's what stops the spread. So I think it depends on the pathogen in question where the cholesterol is playing a role.
Okay, thanks, Aaron. So apologies to those of you asked questions and we don't have time to answer them, but that's the end of today's seminar. So thanks everyone for listening, and thanks very much, Aaron, for a great seminar. Okay.
Thank you. Bye.
Bye-bye. Bye-bye.